Welcome back to BioBiz Buzz. I'm your host, Mike Ward, and today we're exploring one of the most challenging areas in oncology, multiple myeloma. Despite significant advances in recent years with CAR-T therapies and antibody treatments, this blood cancer still remains incurable, and patients continue to face relapse and devastating consequences. Joining me today is Dr. Chris Baldwin, who's the CEO and Managing Director of HaemaLogiX. It's a clinical stage Australian biotech company that's taking a differentiated approach to treating multiple myeloma by targeting antigens that actually current therapies miss entirely. So Chris, welcome to BioBiz Buzz.
Great. Thanks, Mike. Really great to be here.
Chris, let's start with the basics. For our audience who may not be deeply familiar with multiple myeloma, can you paint a picture of the disease burden and why this remains, I guess, such an important unmet medical need?
Yeah, sure. So multiple myeloma is the second largest blood cancer worldwide. It affects plasma cells in the bone marrow. And what makes it particularly challenging is that it's currently incurable. Patients diagnosed with myeloma face a median survival of about 5 to 7 years, and the disease progresses significantly and damages the bone marrow as it does. It causes bones to become destroyed or break, kidneys fail, and there becomes severe immune suppression. The global market to treat multiple myeloma is quite substantial. It's approximately $25 billion a year, and it's growing at 8% a year. But the important point is this, that despite all the impressive innovations that we've seen with the different drug classes like proteasome inhibitors, immunomodulatory drugs, CD38 antibodies, and now most recently BCMA-targeted cell T therapies and bispecifics, patients invariably relapse. And when they do, many lose the expression of the antigens that are being targeted by the immunotherapies, making the treatments extraordinarily difficult.
Yeah, I guess that's sort of the crucial point. Antigen loss at relapse. The BCMA targeted therapies that you mentioned, Idical and there's Cilta-cel, they've generated sort of significant excitement with response rates sort of approaching 80 to 98%. But you're suggesting that their very success creates a selection pressure that actually leads to resistance.
Yeah, exactly. BCMA has been revolutionary as a target. It's highly expressed on myeloma cells, and it's led to some real breakthroughs. But now we're beginning to see the clinical literature that shows that BCMA expression can actually be downregulated or lost entirely following the BCMA-directed therapies. Studies have shown that BCMA low or negative plasma cells can continue to grow and lead to clinical relapse. And in some cases, patients develop complete loss of BCMA through gene deletions. Moreover, the BCMA-targeted therapies, particularly CAR-T, come with a lot of toxicities as well. Cytokine release syndrome is well known. It occurs in 75 to 95% of patients. Importantly, neurotoxicity, severe and prolonged cytopenias, and life-threatening infections also occur in the BCMA-targeted CAR-Ts. Many of the toxicities stem from the fact that BCMA is expressed at small levels, but certainly significant ones, on normal plasma cells and sometimes healthy B cells as well. So you're really training the body to attack the B cell structure that produces the antibodies that protect the patient from infection. So these off-target effects can be devastating to the immune system precisely when patients need it the most to fight the cancers. They face a lot of opportunistic infections along the way.
Yeah, so I guess that sort of brings us to HaemaLogiX's approach. As I understand it, you've identified entirely different antigen targets. So can you walk us through what makes these antigens unique and why they represent a potential paradigm shift?
Yeah. So our scientific founder, Dr. Roseanne Dunn, has made a career after she found a critical discovery, which is that what we call the myeloma antigen that is uniquely expressed on the malignant plasma cells, but basically absent from any healthy cells, allows the body to kill the cancer cell that causes multiple myeloma. What's remarkable is how specific this is, because what's unique is that these antigens arise from the abnormal presentation of immunoglobulin free light chains on the cell membranes of cancer's plasma cells. Normally, healthy plasma cells don't express them at all. What this means is that we can really target tumors rather than the immune system. If you look at plasma cells when they function properly, they're using one of two variants of the light chain protein to produce the unique antibody that nature's developed them to produce. And so kappa variant and the lambda variant produce the antigens on the malignant plasma cells. So when it's in the malignant state, we call it the KMA, the kappa myeloma antigen, or the lambda myeloma antigen. And if you look at the patient base, 70% of the multiple myeloma patients have the kappa type disease and express KMA. The other 30% have the lambda type disease and express the LMA. So our approach is to treat these two patient cohorts separately. The kappa patients would be treated with the antibodies that bind to KMA or KappaMab, and lambda expressing patients would be the ones who are treated by lambda-mab. And what we find is that those antigens, KMA or LMA, are typically higher than BCMA in many cases, which means that it's going to be a very rich and potent target. And that what's really interesting is that as disease progresses and other treatments are applied, those antigens actually increase and enrich under treatment pressure.
Wow. So, I mean, that's fascinating. So, you're potentially solving two problems. You're avoiding the off target toxicity, and you're targeting antigens that persist or even increase under treatment pressure.
Yes, exactly.
Right. Okay. So, I mean, that's interesting. Let's sort of switch now to where you're at with the development of your programs. Your clinical data. Your lead asset, KappaMab, it's a monoclonal antibody that's already generated some Phase IIb results. So, what have you seen?
Yeah, so the data with KappaMab has been really encouraging. We had a Phase IIb trial where we evaluated KappaMab at a 10 mg per kg dosing regimen in combination with lenalidomide and dexamethasone, which is standard backbone therapy, in patients with relapsed or refractory kappa-type myeloma that have received at least 2 prior lines of treatment. And this combination achieved an overall response rate of 83% compared to just 45% in the matched real-world cohort that received lenalidomide and dex alone. And really, even more compellingly, what we saw was a 40% reduction in the risk of death, which was highly significant. And the safety profile was really great. Essentially, it was equivalent to lenalidomide and dex without KappaMab. We didn't see any additional toxicity or immunosuppression with the addition of KappaMab. And these results were published in British Journal of Hematology. And we also presented it, of course, at international conferences, or it wasn't us, but the researchers who conducted the trials, of course. And based on the data, what we're doing now is advancing to a higher dose trial, up to 30 mg per kg KappaMab in combination, this time with pomalidomide and dexamethasone, in order to further optimize efficiency.
Okay, so I mean that's exciting with KappaMab, but as I understand it, you're not actually just stopping with monoclonal antibodies. You also got a CAR-T program as well. So can you sort of tell us about KMA.CAR-T and also about the sort of the partnership that you have with the Peter MacCallum Cancer Centre?
Sure. Yeah, that's right. So we want to be modality agnostic because our expertise is with the KMA LMA target. So whether it's CAR-T or bispecifics or traditional monoclonal antibodies, we can learn a lot about the potency and the capability of addressing this target using either monoclonal antibodies like KappaMab or the CAR-T therapy approach. Our KMA-CAR-T is a chimeric antigen receptor T cell therapy that we've developed in collaboration with Peter Mac, which is, for those of you who don't know, one of Australia's premier cancer research and treatment centers. They have a world-class experience in CAR T development and manufacturing. They've been on the forefront of many of the clinical trials that have been done across the CAR T therapies. And they're conducting the phase I first in human trial of KMA.CAR-T. What got us going on this approach, the CAR T approach, was preclinical data using xenograft mouse models that showed highly selective killing of KMI expressing myeloma cells and very robust anti-tumor activity. So the study that we're about to commence is designed to enroll initially 6 patients, potentially expanding to 12, expecting to see the first patient in probably 2 months, so in the second quarter of 2026. Importantly for us, Peter Mac is going to be the manufacturer for the CAR T cells using their commercial scale GMP facility. So that really de-risks the manufacturing aspects compared to companies that are trying to build that capability from scratch.
Right, 'cause I guess so. That's one of the advantages you have, that partnership infrastructure. You're not carrying the full manufacturing clinical burden yourselves at this early stage, which clearly is important for a young biotech. Let's talk about your pipeline beyond KappaMab and KMA.CAR-T. You mentioned lambda for the lambda type patients. So what's the sort of development status there?
Yeah, lambda is really interesting for us because it's expressed on 30% of myeloma patients. We're very interested in it. It has a strong, very large potential addressable market of its own in the cancer space. And the preclinical data that we've seen with it is very consistent with KMA. Obviously, as a small biotech, we've been focused on lambda with the larger market and the higher population pool of patients, but we need to lift the LMA up into clinical trials as well. Because if we see the same sort of response, highly selective binding to lambda plasma cells in this case, and no off-target binding to normal tissues. But what's also interesting about LMA and KMA are they're expressed in other diseases, including AL amyloidosis, which is a rare, often very fatal disease where abnormal lambda light chains are deposited in organs and cause organ failure. There's a huge unmet need in AL amyloidosis. And unlike myeloma, where the majority of patients are kappa type, AL amyloidosis, they tend to be lambda type. And in fact, our data suggests lambda is in about 90% of AL amyloidosis cases. So progressing lambda is not only important for us, but it's very important for patients that are facing AL amyloidosis.
Okay, so let's shift gear and let's talk about the business and strategic landscape. As I understand, you're preparing for an IPO on the Australian exchange sometime this year, and you're targeting a raise of $25 million based on recent reports that I've seen. Can you help our audience understand what are the key value inflection points that you will need to hit to make this a successful public company? And what are the significant risks that you're having to manage?
Yeah, great question. So we have 3 strategic priorities. First, we need to successfully initiate and complete the enrollment in that higher dose KappaMab Phase IIb trial study that we discussed. This is going to provide the dose escalation data we need and also help us define the dosing regimen for the potential registration studies. Completing this trial would really position KappaMab for partnering discussions or potentially a pivotal trial design. And secondly, we're focused on that KMA.CAR-T study. We need to treat patients with the Peter Mac in the phase I trial to generate further safety and efficacy data. We think that the early signals from that CAR-T program could really reshape valuations along the way. And that if we can demonstrate the KMA.CAR-T delivers efficacy compared to the existing CAR-T treatments with BCMA and come with a safer profile, particularly in neurotoxicity and prolonged cytopenia, but also potentially the CRS, then we would have a really interesting result. Then third, we need to really advance that lambda mab approach towards IND-enabling studies into the clinic, because this broadens the addressable market and demonstrates platform versatility, as well as, of course, expands beyond myeloma into other disease areas where the KMA-LMA antigen is relevant.
Yeah, and you're thinking about the risk. I mean, you're operating in what's clearly an extraordinarily competitive space with multiple BCM targeted therapies already approved, and we're seeing dozens more in development. And of course, there are also other, I guess, alternative targets as well. So the GBRC5D and the FCRH5, which are gaining traction. So how are you going to differentiate yourself in such a crowded market?
Market dynamics are competitive, no doubt. I'd reframe the challenge a little bit, because if you look at it, there are, especially with BCMA, there are a lot of therapies that are coming there, as well as the emerging targets. But really, the clinical reality is that patients do exhaust the options that are there and the options that are being put out. Clinicians are really interested in new targets despite the progress with CD38 and BCMA. BCMA refractory myeloma is becoming really a significant clinical cohort because of their success. So these patients desperately need alternatives as they relapse and become refractory. Our differentiation, when we talk to clinicians at Mayo Clinic or at Dana-Farber in Boston, they point to three distinct areas. First, with KML-LMA, of course, this is a novel target no one else is working on to the extent that we are. We've invested very heavily in the IP around it, and that IP position is going to be critical for us. Secondly, the safety profile suggests that we can really avoid the severe toxicities that limit other therapies that are existing now, which means better patient eligibility, potentially earlier lines of therapy, longer periods of time on treatment, and ultimately, fundamentally, better quality of life for patients. And third, as we mentioned earlier as well, our data suggests that the targets that we're going after may actually be upregulated for heavily treated patients, which is pretty exciting, because often when you're dealing with a heavily treated patient, the options are exhausted and drugs, frankly, start to work worse and worse. So we're potentially best positioned where other therapies are failing. The risks that we have to manage to execute on this really involve the clinical trial execution, making sure we get patients enrolled, managing trial timelines and what have you. And then, of course, there's always going to be with CAR-T manufacturing risk, because every patient gets their very own batch, of course. That collaboration and the incredible experience of Peter Mac really mitigate a lot of that. There's always the issues of regulatory risk, but we've designed these trials with regulatory input. We have two scientific advisors who both developed drugs that exceeded 100 billion in worldwide revenue over their journeys. So we've really leaned heavily onto their experiences as we've developed these trials, and we're following on well-established development pathways. But of course, the top of mind for me right now, where we are in the journey, is the capital risk. We need to secure sufficient funding to achieve the inflection points. We don't get to do this for free, unfortunately. And biotech funding has been improving a lot in the last year or two, but naturally, it's always a bit selective.
Yeah, sure. I mean, capital risk, that's definitely going to resonate with the biotech execs who are listening to this podcast. As you said, sort of the capital markets have been sort of a little bit lackluster, since 2023. We saw sort of the end of 2025 and early in 2026. We've seen some, I guess, signs of a thaw. But how are you thinking about this capital strategy? I mean, is the IPO your primary path, or are you sort of, maybe considering strategic partnerships, non-dilutive financing, or even other structures?
Yeah, I think, like, as you said, like any other biotech in this space, the answer is going to be all of the above. The ASX IPO is our primary near-term objective, but really that's the table stakes, right? That's what gets us in the door, gets the KappaMab study launched, gets the results and the readouts from the Peter Mac trial, as well as helps us develop the lambda mab. It's the capital that provides the runway to generate all that meaningful clinical data that we think will give us the inflection points we need. The next step is going to be exploring the partnership opportunities, especially with KappaMab, given where it is in its journey. A partnership with a larger pharma or specialty oncology company is a traditional pathway for drugs in oncology such as this. And that would provide a lot of upfront capital to fund the late stage development. And obviously, on the back of that, with success is what drives the commercial infrastructure necessary to bring the drug to patients. Given the data we've seen in the Phase IIb thus far and the one we're about to do, we think that KappaMab is highly partnerable and potentially synergistic with some of the bigger players. Non-dilutive funding, of course, is always on the table with government grants and collaborations. There's also disease foundation support, especially with ALM amyloidosis, where patient advocacy is strong and unmet needs are profound. So the answer is yes.
Yeah, I'm just thinking again, it's like, the risks. Manufacturing, particularly sort of cell therapies, that this sort of CAR-T manufacturing is notoriously complex, expensive, and these days capacity constrained. I know you've got the Peter Mac partnership, but that's for phase one. How are you sort of thinking about manufacturing scale up? For example, if KMA.CAR-T actually succeeds, are you going to build internal capabilities or partner with a CDMO or pursue co-development and commercialization partnerships?
Yeah. So obviously, like you said, manufacturing is a big strategic decision that we face, and CAR-T manufacturing is notoriously complex and unique with respect to the fact that it's often done near the patient and in these kind of one patient batches. The manufacturer requires specialized clean rooms, skilled personnel, the supply of the viral vectors that turn the patient's T cells into CAR cells, and really requires a lot of logistics. So the capital investment there is pretty prohibitive for a company like ours. But most likely, we'll be looking where to find an established CDMO that has that capability and expertise, geographically diverse locations. What you see now and what's really becoming important to the industry, especially in multiple myeloma, is that some of the larger pharma companies have established that CAR-T capability themselves to bring their products, cell-to-cell as an example, to market and can solve that manufacturing challenge with the commercial strength, of course, that they normally bring to everything. You can also see a lot of changes in the way CAR-T is done with new modalities coming up. There's change in structure, there's change in technology that make CAR-T better in terms of its ability to reach patients in diverse locations, and also starts to dramatically change the economics and accessibility for CAR-T. So we're obviously very happy to explore that.
Yeah, I mean, it's interesting that you mentioned it's like, patient access and affordability, because in many respects, again, when we're sort of talking about, things like cell therapies, it is the elephant in the room, the patient journey and market access. So, even if you get regulatory approval, reimbursement and market access can still be a significant hurdle. So how are you thinking about health economics and pricing and payer strategies?
Yeah, it's a really good question, and it is pretty different based on the different modalities that are out there and how used to antibodies or how used to CAR-T some of these economic systems are built. So the health economics argument will ultimately be based on the comparative effectiveness and safety. If we can show the KappaMab combined with standard of care delivers a superior survival with equivalent or better safety, which often comes at a better cost compared to standard of care alone, then you see that cost effectiveness become really compelling. Payers are increasingly focused on value and value-based pricing. They want to see survival benefit and quality of life rather than simply extension of life and reduce total cost of care, including hospitalizations and supportive care, become important in those calculations. For CAR-T, the challenge is always going to be greater due to the cost and the complexity, but the arguments are the same. If you can deliver something that provides durable remission, reduces hospitalizations, and provides longstanding relief to basically incurable disease, the total cost of care can potentially be lower with existing CAR despite the big upfront. And again, you can see a lot of technology providers working on solutions for that high upfront cost problem associated with CAR-T. So we're exploring all the different approaches, the contracting models, finding ways to look at outcome-based agreements where pricing is tied to response or survival metrics. Several of the EU countries and US payers are increasingly open to structures like that.
Clearly big challenges though. So as we draw to a close, I'd just like you to sort of share with us your thoughts of how HaemaLogiX, how you sort of see it sort of like developing over the next 3 to 5 years. What's success going to look like for the company? And I guess perhaps more philosophically, what impact do you hope to have on the patient lives?
What often strikes me about cancer therapies is just the number of pills that a patient ends up walking out of the hospital with, and all the drugs that they end up having to take to protect and ameliorate the off-target effects or the side effects that the primary drug causes. So we're really motivated to make life better and longer lasting and easier for those patients. So for us, success in 3 or 5 years means that we have at least one of these assets, probably KappaMab, in a pivotal trial, potentially partnered with a major pharma company moving towards registration. Because it would mean we have clinical proof of data. We have shown already through the phase IIbs of the power of the drug, and we're really trying to make sure that we can deliver something that's going to affect people's lives. Success in the next 3 to 5 years also means that we have clinical proof of concept data for KMA.CAR-T, demonstrating safety and efficacy signals there. That kind of information about the different modalities on the same target can be incredibly powerful and really home us in on the right approach to treating myeloma. And it would also mean that we don't leave the other 30% of myeloma patients behind that have the lambda type disease, that we've moved the lambda through into the pipeline. It's into the clinic. We're getting phase I results, and we're getting these next generation constructs prepared. Ultimately, of course, what we're really hoping to achieve is success measured through patients' lives. Multiple myeloma it's a devastating disease. The treatments and the lifestyle that patients endure over the journey, the repeatedly relapses, the accumulation of different toxicities and debilitations are really heartbreaking. And if we can offer a therapy that extends the survival, particularly as it improves the quality of life, allows the patient's immune function to persist, and allows them to spend time with families, travel, and work, that's really what drives us. The science that Roseanne and the company have been working on for some time is really something special. This opportunity from a business perspective, obviously, is incredibly large, but fundamentally, the mission is human. We're trying to give patients more time, better time. It's what motivates us. And we're really excited about the next step in our journey.
Well, Chris, I think that's a very powerful note to end on. So thanks so much for joining us on BioBiz Buzz and for sharing the HaemaLogiX story. I mean, it's clear you're tackling one of oncology's toughest challenges with differentiated science, strategic partnerships, and a clear-eyed view of what that road ahead looks like. So we'll be definitely watching closely as you sort of progress through sort of the clinical development and towards your IPO.
Great. Thanks, Mike. It's really been a pleasure.
And for our listeners, thank you for tuning in to BioBiz Buzz. If you found this conversation valuable, please subscribe, rate, and actually share it with colleagues who you think would benefit from hearing about innovation at the cutting edge of cancer immunotherapy. Until next time, I'm Mike Ward, and keep on innovating.